CareDx is a transplantation-focused company dedicated to developing the best products to improve outcomes for transplant patients. Better matching and better surveillance equals better outcomes. Today, we're going to talk about AlloSeq cell-free DNA, CareDx's kit-based solution that enables measurement of donor-derived cell-free DNA through a simple blood test, making it an excellent post-transplant surveillance tool. In this video, we're going to show you how easy it is to use and implement this assay in your lab. The instructions for use, also known as the IFU document, will also showcase the detailed procedure of what we are going to describe. Please read the document to understand the limitations and the contraindications as well as other considerations before using AlloSeq cell-free DNA in your lab. Okay, so let's begin. We start with sample collection. We recommend drawing blood into 2 Streck cell-free DNA BCT tubes per patient. These blood collection tubes are designed to stabilize nucleated blood cells to allow for high-quality cell-free DNA extractions. The next step is to isolate plasma according to manufacturer's instructions. We recommend using the double spin protocol to isolate the plasma. Then using the instructions as provided by QIAMP circulating nucleic acid kit from QIAGEN, extract cell-free DNA from plasma. Next, quantify the input DNA before beginning the protocol. We recommend using a fluorometric quantification method such as Qubit or picogreen, according to the manufacturer's instructions. We recommend that input of 10 nanograms of DNA per sample be used with the AlloSeq cell-free DNA kit. Dilute the DNA in RNase-DNase-free water so that the volume of DNA sample input per well is 60 microliters. So now you have your extracted DNA. We will continue with library preparation. The first step in the protocol is to set up a multiplex PCR. The following reagents are needed for PCR setup: AlloSeq cell-free DNA PCR mix, AlloSeq cell-free DNA PCR enzyme, AlloSeq cell-free DNA SNP primer pool, AlloSeq cell-free DNA index primers that include forward I5 and reverse I7. Since these reagents within the kit are stored at -15 to -25 degrees Celsius, first thaw reagents at room temperature and then place on ice. Keep reagents on ice during setup. The PCR program should be set up for a reaction volume of 40 microliters. It requires specific ramp rate settings for the first 8 annealing at 57 degrees Celsius and elongation at 72 degrees Celsius steps. The ramp rate equivalents for different thermocyclers and PCR cycling protocol are provided in the tables within the instruction for use document. Let's get into the process now. Dilute sample DNA in nuclease-free water to 10 nanograms in a final volume of 16 microliters. In a PCR plate, on ice, add 4 microliters of the forward index primers, I5, to the sample wells, based on the plate layout defined in your sample sheet. Keep track of which primers are being added to each well of the plate. Add 4 microliters of the reverse index primers, I7, to the sample wells. Add 16 microliters of your prepared DNA to each sample well. Now let's focus on the master mixes for PCR. Prepare a master mix in a 1.5 ml Eppendorf tube for your samples using the table mentioned in the instructions for use. I have already prepared this using the table here. Vortex this master mix for 5 seconds to mix and centrifuge briefly. Add 16 microliters of the prepared master mix to each sample well. Apply microseal B and immediately vortex the plate for 5 seconds to mix. Immediately centrifuge plate at 1000 Gs for 30 seconds. Check that no bubbles are present. Immediately place the plate on the thermocycler in a post-amplification area and run the PCR cycling protocol. The PCR cycling protocol takes approximately 1 hour. What happens during this reaction? Let me show you how this assay works. At its core, AlloSeq cell-free DNA is a targeted next-generation sequencing assay that utilizes differences in single nucleotide polymorphism loci to measure the amount of donor-derived cell-free DNA relative to the total amount of cell-free DNA derived from a plasma sample. After a few rounds of specific primer amplification, additional primers present in the reaction containing index and adapter sequences anneal to the target specific product, generating sequencing-ready amplicons, that is, All 202 amplicons in the same well will contain a single set of sample-specific dual barcodes and Illumina-compatible adapters. The kit comes with a total of 4 I5 forward and 6 I7 reverse index primers that can be used for up to 24 unique combinations. You can multiplex up to 24 samples with various index combinations. And that's how the assay works. Coming back to our protocol, now that our PCR process is complete, we pull our libraries, clean them up, and quantify them before sequencing. For Pcr Product Cleanup, you will need AlloSeq cell-free DNA purification beads and AlloSeq cell-free DNA resuspension buffer. Before you begin, make sure that you have removed the purification beads from 2 to 8 degrees Celsius storage and equilibrate to room temperature for at least 30 minutes. Also remove resuspension buffer and PCR plate from 2 to 8 degrees Celsius and equilibrate to room temperature. Centrifuge at 1000 Gs for 30 seconds. Prepare 80% ethanol, 3 ml total. Now let's mix the AlloSeq cell-free DNA purification beads and the PCR product. First, you need to determine the volume of each sample to pull into a final volume of 120 microliters as in the examples in the tables as shown in the IFU. For today's video, we are working with 2 samples. However, if you are working with 10 samples, you'll pull 12 microliters per sample into a 1.5 ml Eppendorf tube. Add 100 microliters of AlloSeq cell-free DNA purification beads to the tube of pulled samples. Vortex the tube for 5 seconds on full speed and incubate tube for 5 minutes at room temperature. Place the tube on a Dynamag 2 stand and allow beads to pellet for 5 minutes or until the liquid is clear. Keep the tube on the magnetic stand and remove all supernatant. After this, we wash twice with 80% ethanol, making sure to not lose any beads during the process. Remove the tube from the magnetic stand and add 35 microliters of the AlloSeq cell-free DNA resuspension buffer. Vortex the tube for 5 seconds on full speed and incubate tube for 5 minutes at room temperature. Place the tube on the Dynamag 2 stand and allow beads to pellet for 5 minutes, or until liquid is clear. Without touching or disturbing the beads, transfer 32 microliters of supernatant using a smaller displacement pipette to a new 1.5 ml Eppendorf tube. Your libraries are now ready to be quantified and diluted before sequencing. Quantify it as per your usual methods. Dilute and spike in 1% Phi-X if required, and it's ready for sequencing. That's it for the hands-on protocol. You then let the sequencing run and analyze the data using AlloSeq software. Please refer to the instruction for use document to learn in detail about this protocol, but as you just saw, it is easy to use and adapt in your own labs. If you have any questions, please reach out to your local CareDx representative. Thank you for your time.